My name is Ricardo Herpal, and I'm representing the LatinX group at Shades. So LatinX at Broad has the mission of building community among Latinx Broadies and allies and empowering members of the Latinx community. We want to highlight Latinx and diverse researchers within the Broad and outside the Broad. We want to foster collaboration, and we want to allow visibility and role models for diverse individuals. We also want to promote research in our community and opportunities to network. So if you would like to join, if you'd like to help out, please send Latinx at the Broad an email, or you can Slack me directly, and we can continue this speaker series. So today I would like to introduce and present to you a former Brody, Amanda Tomi Ouchida. She's a Brazilian biochemist with 10 years experience in cell death, cancer, and development of high throughput screening assays. Amanda got her PhD in biochemistry at the Universidad de Sao Paulo with an internship at Harvard Medical School. After 2 years of postdoctoral studies, one at the Karolinska Institute at Sweden and another at the Albert Einstein College of Medicine, New York, she worked as a scientist at the Broad Institute in the Center for Development of Therapeutics. During her time working at the Broad, Amanda worked in several projects at early stages of drug discovery, focusing on the development and optimization of assays for target validation and mechanisms of action, elucidation of these mechanisms. Recently, Amanda moved to pharma, and she is working at Takeda Pharmaceuticals as a scientist in the toxicology department. So I'd like you to welcome Amanda once again.
Thank you very much for having me here today. For me, it's an honor to come to the LatinX talk because when I was at the Broad, I was part of the LatinX group. So for those that are not part of this group yet, I highly recommend you to join. It's very nice. It gives us a lot of support as Latinos. So what I'm going to speak today is how we can modulate cell death as a path for disease treatment. So I know that Ricardo already gave an introduction, but I would like to just say who I am again, because initially we were going to do this talk as like a career development talk. So if you have any questions regarding that aspect, I can answer those as well. So I'm a biochemist. I have a bachelor in biochemistry by Universidade Federal São João do Rei, back in Brazil. My PhD is also in biochemistry by Universidade de São Paulo. And there I start to study the mechanism of cell death resistance in cancer. During my PhD, I came to Harvard Medical School to develop some research during 1 year. That's the work I'm going to show here today. And there I study some autophagy inhibitors that could be used to the cancer treatment. After my PhD, I went to Karolinska Institute and I was a postdoc there working with a specific type of autophagy that's called chaperone-mediated autophagy. After that, I did a second postdoc at Albert Einstein College of Medicine, and there I was trying to find some protacs that would target bugs and that could be used to attenuate chemotherapy-driven cardiomyopathy. At the Broad, I was at CDOT, and I was working basically with assay development for high-throughput screenings. I was very happy at the Broad, I can't deny. I miss the Broad sometimes. And now I'm at Takeda. I'm a scientist too at investigative toxicology. And what I do here, I try to develop assays that are going to de-risk potential toxicities that come from different modalities that we have here at Takeda. So I'll divide this talk in two parts. So the first one, I'm going to focus on the work that I did at Harvard Medical School during my PhD. And the second one is a short one that I did during my postdoc at Einstein. But I just brought these two because I want you to see the two different ways that we can modulate cell death and induce in a therapeutical way, let's say like that. So the first one, we are going to speak about autophagy modulation as a strategic approach to induce cancer cell death. So Hanna and Weiberg have described 10 characteristics that a cancer cell needs to have in order to be considered malignant. And one of these characteristics is the resistance to cell death. So this group in Germany, they did an RNA-seq of 7 breast cancer patients, and then they tried to capture what was the most mutated pathways that they could observe in those patients with cancer. And as you can see here, apoptosis signaling is one of these mutated pathways. So mutations in the cell death signalings can lead to cell death resistance in cancer. But how about autophagy? So we have described so far more than 20 different types of cell death. And autophagy is also considered one different type of cell death. There is some contradictions in this aspect, because some researchers consider autophagy as a cell death pathway and others don't. But in any case, what is autophagy? Autophagy is the degradation of certain damaged organelles or misfolded proteins through the lysosomes. So how does this process work? So when in conditions of starvation or other types of stress, we have the damage of some organelles and also some unfolded proteins. This will lead to autophagy induction. Autophagy induction in general, it's done through mTOR inhibition. And the way that autophagy is triggered is by initially we have the isolation of a double membrane. This membrane can come from the ER, can come from the mitochondria sometimes. So it depends the source of the membrane. So after this first step of the isolation of the membrane, we have the vesicle elongation, where we have the phagophore formation, and then we start to sequester the cargo that's going to be degraded. Once we have the formation of the whole vesicle, this structure is now called autophagosome, and we have the docking of the cargo in the double vesicle membrane. When we have the fusion of the autophagosome with the lysosome, we then have the structure called autolysosome, and then the proteases existent inside of the lysosome are going to degrade the cargo. And this process we call autophagy. So far, there are three major types of autophagy that can be described. We have macroautophagy, microautophagy, and chaperone-mediated autophagy. And the main difference between these three different types of autophagy is the way that the cargo is delivered to the lysosomes. So in the case of the macroautophagy, we have the formation of the autophagosome. That's the process that I just described. And this macroautophagy can be selective, so certain different types of cargo. So we have mitophagy, that's the degradation just of the mitochondria. We have ribophagy, that's just of the ribosomes. And we can also have macroautophagy in bulk. That's when we have no distinction of what the cargo is. So for microautophagy and chaperone-mediated autophagy, we have the delivery of the cargo directly to the lysosomes. And these two types of autophagy, they are more selective. In this case, we have HSC70, that's a chaperone, that's going to help this protein to be delivered to the lysosome. In the case of the chaperone-mediated autophagy, we have the presence of two different proteins. I'm going to mention them again later on in this talk. I just want you to remember that one of them is LAMP2A, that's a transmembrane lysosomal protein. And again, we have the help of the HSC70 chaperone to deliver this cargo to the lysosome. And autophagy has a dual role in cancer. So it can either promote tumor growth. So the overactivation of autophagy can kind of create a cytoprotective mechanism, helping these cells survive in a low nutrient condition. But on the other hand, if autophagy is induced in the early stages of the tumor development, it can act as a tumor suppressor by removing damaged organelles and proteins and also reducing the chromosome instability. So based on that, we can say that we can target autophagy to induce cancer treatment. So you can either inhibit autophagy by inhibiting autophagy, you can stimulate apoptosis. That's a different cell death pathway. On the other hand, you can boost autophagy and induce autophagic cell death. So many autophagy inhibitors are already described in the literature, and most of them, they target different stages of the autophagic process. So 3-methyladenine, for instance, is a PI3 kinase inhibitor, and it's going to target the autophagosome formation, so in the beginning of the pathway, but is not very well recommended because the PI3 kinase that is involved in autophagy is a PI3 kinase class 3. So when you use 3-methyladenine, you're not just going to inhibit PI3 kinase class 3, but also other types of PI3 kinase. A different type of autophagy inhibitor that I want to describe here as well is spaltin-1. Spaltin-1 acts in a different way. I'll speak more about it 2 slides later. But the way that spaltin-1 acts is by blocking the inducing the degradation of backlin-1. So if we don't have backlin-1, we are not going to have autophagy induction, because backlin helps in the initiation of autophagy for the phagophore formation. And we also have chloroquine and hydroxychloroquine that are going to act in the last step of autophagy. That's the fusion between the autophagosome and the lysosome. So it can either impair the fusion of both, or can also basify the lysosomes, and then by increasing the pH, the proteases inside of the lysosome cannot work, and then we don't have the degradation. So we have in clinical trials right now different chemotherapeutics in combination with hydroxychloroquine. That's this example here. So in general, it's common to combine an autophagy inhibitor with a chemotherapeutic to reach cancer treatment. But why use the combined therapy instead of using the single therapy? So if we look at this scheme here, for many times, if you treat a patient with a drug A, after a while, this patient can be resistant to this type of drug. And by the time you add the drug B, you're going to have 2 different cell populations. One that's resistant to drug A, another one that's resistant to drug B, and one that's resistant to both. If you start the drug combination since the beginning of the treatment, you're going to avoid this type of resistance mechanisms. And also, you can target different pathways in the cell, making your treatment more effective. So AC220, or quizartinib, it's a potent second-generation FLT3 inhibitor that's in phase 2B of clinical trials. And it has been proven that this compound, it's better when used in a combination therapy instead of a single agent. So when I joined the lab during my PhD, the lab at Harvard Medical School, they had just found spaltin-1. Spaltin-1 here is shown as C43. So they have shown that C43 was an autophagy inhibitor. As you can see here in this figure, they use LC3 as an autophagy marker. So LC3 is a protein that's lipidated during the autophagosome formation. So if we have more LC3-2, this means that we have more autophagy activation. Rapamycin is an mTOR inhibitor. So once we inhibit mTOR, we have more, we have autophagy stimulation. And when we combine rapamycin plus C43, that's spaltin-1, we have less LC3-2, meaning that we have autophagy inhibition. So since they had found the spaltin-1, they run a screening in the lab to try to find a drug that in combination with the spaltin-1, they were able to induce cancer cell death. And this compound was AC220. So as you can see in these 2 figures, when they combine C43, that's spaltin-1, with AC220, they could induce cancer cell death. So we know that this combination is able to induce cancer cell death, but when they went to in vivo studies, they observed that spalting 1 by itself caused cardiotoxicity. So my job when I joined the lab was trying to find a new autophagy inhibitor that in combination with AC220, we were going to be able to see cancer cell death. So for that, I run a small molecule screening to try to find autophagy inhibitors. I use ES2 cells, that's an ovarian cancer cell line, and I run several libraries in a total of 12,000-ish compounds, and I run viability assays using cell titer glow as a readout. And I had always two sets of plates, one with quizartinib and one without quizartinib. And when I was selecting those compounds as hits, for me was important that whatever compound I found, it was inducing the cell death or was increasing the cell death just when in combination. So out of my primary screening, I found 45 primary hits. And those primary hits, they were further tested again in viability assays. And also include the assay that are going to see if they are an autophagy inhibitor or not. That's a GFP-LC3 assay. So what is this assay? So basically, you generate stable cell lines that express DLC3, that's that autophagy marker that I told before, conjugated with the GFP. And when we have autophagy activation, we have puncta formation in the cells because this GFP-LC3 can then go to the cell to help in the autophagosome formation. So I run the GFP-LC3 assay as a secondary screening, and I found TAC165 or muberitinib, that's a HER2 inhibitor, as a hit. When I further validate TAC165 in combination with AC220, as you can see here, I could observe cell death when I treat with the combo, but not when I treat with the compound alone. I run these assays, the screening just with this ovarian cancer cell. So for me, it was important also to see if other types of cancer were also affected. So as an example, I'm bringing one type of breast cancer cell line. In total, I tested 10 different types of cancer cells, but here I'll show just these 3. That's a breast cancer example, that when we treat with the combination, we have the decrease of the viability. One AML example, that again, the combo was able to induce cancer cell death. In a triple negative breast cancer cell, and this one is interesting because a triple negative breast cancer cell is a cell that doesn't have HER2, and TAC165 is a HER2 inhibitor. And even in this type of cell, I could observe a more pronounced cell death when I induce when I treat with the combo. But the question was still that I need to validate if TAC165 inhibits autophagy. So I repeat again the GFP-LC3 assay. And as you can see here, we can see more puncta with rapamycin, and this puncta reduced after TAC165 treatment, being an indicator that TAC165 can inhibit autophagy. However, in autophagy studies, just the reduction of LC3 puncta is not enough. You need to see if the autophagic flux is blocked. So the flux means since the autophagosome formation and the dock of the cargo until its degradation. So we need to see if this process is being paired or activated. And by doing that, in general, what we do, we use chloroquine as a tool to block the last step of autophagy. So as you can see here in this Western blot example, when we starve cells, in general, we have more LC3-2 formation, as we can see here. And when we block these with chloroquine, we are supposed to see even more compared with the chloroquine alone control. So I run a similar assay. So I starved the cells with a glucose-free media, and I did glucose-free plus chloroquine. So we have here an increase of LC32. When I treat the cells with TAC165 in the same glucose-free conditions plus chloroquine, I could not observe the LC32 anymore, showing that TAC165 can really impair the autophagic flux, being an autophagy inhibitor. So we know that TAC165 is a HER2 inhibitor, and what I'm observing is autophagy inhibition. Then the question was, does TAC165 inhibit autophagy through HER2, or is it through an off-target effect? To address that question, I generate HER2 knockdown cell lines using shRNA technology. And at that time, we didn't have CRISPR, guys. I'm sorry. And as you can see, I could perform the knockdown of the cells. And again, I starve the cells in a glucose-free condition. You can see in the control cells that receive the scrambled shRNA that we have more LC32. However, in the knockdown cell lines, in the glucose-free conditions, I could still observe the LC32 there, showing that by removing HER2, I could not see autophagy inhibition. And most interestingly, when I treat the cells with the combo TAC165 and AC220 in the two different clones of HER2 knockdown that I had, including the scramble control, I could observe cell death in these conditions, but not when I was just treating the cells additioning the AC220. So this is a proof that TAC165 can inhibit autophagy, but is in a HER2 independent manner. Then the next question was, what type of cell death this combo is inducing, right? So as I said before, we have more than 20 different types of cell death described so far, but the most studied ones, let's say it's autophagy as well, but apoptosis and necroptosis. So in order to check if these were the mechanism by which the cells were dying, I used ZVAD, that's a pan-caspase inhibitor, and these three different necroptosis inhibitors. And as you can see in these 4 bars here, when I treat the cells with the combo plus these inhibitors, I could not see rescue of my cell death, the cell death I observed. Caspase-3 and PARP, they're also known as markers for apoptosis, and in general, we see cleavage of caspase-3 and PARP when we have apoptosis activation. In this case, I couldn't see with the after combo treatment known of the caspase cleavage or the PARP cleavage. So this is an indication that this combination does not induce apoptosis, neither necroptosis. However, in that paper that they had found AC220 in combination with spouting 1, they also could not observe rescue using NEC1S or ZVAD, that's the necroptosis and apoptosis inhibitor. However, when they knocked down LAMP2A and HSC70 that are CMA players, they could observe rescue of the cell death. So just as a recap, how does chaperone-mediated autophagy works? So in conditions of starvation or stress, certain cytosolic proteins, they are going to expose a motif that's this QFERQ motif. Once this motif is exposed, the chaperone HSC70 and some other co-chaperones can come and recognize this motif, and they are going to bring this protein to be degraded in the lysosomes. At the lysosomes, LAMP2A dimerizes, it forms a tunnel. Then HSC70 can then deliver the protein for degradation into the lysosomes. The lysosomal HSC70, we have a version of that inside of the lysosomes, is going to help this protein to come through the LAMP2A. And then once the protein is inside, we then have the action of the proteases and the degradation of the protein. So then the question was, does my combo of TAC165 and AC220 is inducing chaperone-mediated autophagy? So in order to see if the combo was inducing CMA or not, I checked the degradation of the main CMA substrates, and those are the mutant P53, I-kappa-B, GAPDH, and exokinase 2. And if we look at the 16-hour treatment with the combo, we can observe the degradation of most of the CMA substrates. And when I knock down HSC70 and LAMP2A, you can observe the rescue of P53, HK2, and GAPDH upon the combo treatment. I also run a viability assay to see if the knockdown of HSC70 and LAMP2A would rescue upon combo treatment. And as you can observe, we can rescue the viability of these cells using both knockdowns. So this shows that TAC165 and AC220 in combination can activate CMA and induce cancer cell death. So as a summary for this part, we have that TAC165 can inhibit autophagy through a HER2-independent mechanism. So we found TAC165 as a new autophagy inhibitor that in combination with AC220 that has been shown before that it kind of mimics a glucose starvation mode, as you can see in these two figures here. They decrease the glycolytic capability of the cell. These two compounds in combination, they can activate CMA, promote exokinase 2 degradation, as we could observe in the previous data, induce metabolic catastrophe, and then induce cancer cell death. So in this example that I just showed, we tried to activate cell death in order to potentially treat cancer. So with that, I would like to thank the labs that I did this work, Dr. Juning Yuan, that was my advisor at Harvard Medical School, and Dr. Evris Gavatiotis and Richard Kitsis that gave me the opportunity to develop this work at Einstein. And if you have any questions regarding this presentation and also career development, just let me know. And I think that's it.
Thank you so much, Amanda. So I'm going to open the floor for questions. So please feel free to ask any questions in the Q&A, and we can unmute you if you'd like to speak. I have one scientific question for you. So, you touched on cancer and you touched on cardiomyopathy, but are there any other disorders that you think autophagy might play a role? Because I know it's a target for neurodegenerative disorders.
I was going to say that.
Yeah.
Yeah. So Ana Maria Cuervo, for instance, that was, I call her the queen of CMA because she was the one that discovered CMA. She has several works showing that CMA plays a role in neurodegenerative diseases. And she even has a startup company that's together with my previous boss, Evers Gavatiotis at Einstein, where they're trying to develop CMA inhibitors and also activators in order to try to cure these neurodegenerative diseases. And I think for some eye problems also, I forgot the name of the disease, CMA also plays a role. So if they find, I think they had a recent publication. To be honest with you, I stopped following CMA, I think 2 years ago, but I believe they have a publication saying that they could find some autophagy inhibitors, CMA inhibitors that could be used to treat those type of diseases.
Thanks a lot. So I put a survey in. So please fill out the survey if you have, so we can improve on the speaker series. And if you know of anyone or you think you would be a good fit for the speaker series, please let us know. Amanda, can you talk about leaving the Broad and going to industry?
Yeah.
That's always always a question for people.
Yeah. So I think nowadays we know that we can do more with our PhDs than before, at least from my time when I started to do my PhD. When I started doing my PhD, I wanted to be a PI. I want to have my own lab and run research and do everything. So during my postdoc, I realized that that was something that I don't want to do, because that cycle of like, you publish to get a grant, and then you do that over and over, and then in the end, that freedom that they speak in academia, at least it's my view, okay, the freedom that they speak in academia, that they have to do research, I think that's not true. In the end, you go accordingly with the trend because you need the grant to run your lab. So I said, I don't want to be part of that. I want to try to do something that's going to help people, help patients. That was my goal. Then I joined CDOT, and CDOT is kind of the biotech of the Broad, where you're exposed to so many different projects and also science. Like I'm still very passionate about the Broad and what we do at CDOT. But however, was not still the pharma thing. So I wanted to experience the pharma. So when I got this opportunity at Takeda, I saw that that could be the next step for me. Like in my mind, that would bring me more close to help people and to help patients. And right now I can see this impact, because working in the toxicology department, I can see how my work can really affect the pipeline of the company itself. So that for me, it's important, and I feel gratitude for that. But to leave the Broad for me was tough, because I really love the Broad. I think the type of science we have there, it's something like amazing, you know, like those conversations that you have in the corridor, like with ideas and like new technology, that thing is science. And here I find that things are more like straightforward. They don't give me this like time to, you know, to be a scientist and think about the problem. I don't know if I answered your question, but that.
I mean, it's always a personal choice, right? It's
Yeah.
always this dichotomy between industry versus academia, and there's more and more spaces I feel where it's kind of blurred between industry and academia. As time progresses and as research changes. Do you think you have more freedom in industry than you did or you would in academia?
No, definitely not. So even though I didn't like the cycle, here I don't have freedom. I don't feel the freedom, you know, because you need to answer a question, because they need the answer to that question. So it doesn't give me time to wonder other things that I would like to explore. You know what I mean?
And is the question always very specific to like the goal? And or is it, I guess depending on where you are, it can be more translational, depending on the question.
I don't know if it's something that I just experienced here at this department, or if in another types, like in another departments, people would have a different view from the one I have right now. But speaking just about the work that I do here, I can say that I don't see much of this freedom, but I can see the translational part, as you said, because I can see that the work I do kind of have impact. I can try to, let's say, they are developing a new medicine now, right? But before putting that in people, they want a sign if that is okay or not, or if you're going to see any sign. So that's what we do here. So even before going to the patients and maybe going there and say that, hey, this thing has a problem, we can kind of hazard ID first. So, hey, maybe not good to bring it now, go back, do some changes, and then maybe after that you can move forward. So I think that's the cool part.
All right, well, unless we have any more questions, I'd like to thank Amanda once again for coming. It's always nice to see a former Brody. I used to work with her actually in LatinX. So thank you so much for coming, and we'll be in touch.
Sure. Thank you very much, everybody. And thank you, Ricardo. I miss everybody at LatinX and at the Broad.